Today, we finally get to what I've been building up uh, for the past few weeks about. And I want to talk about two processes called transcription and translation. The first part is a bit of a review. We covered it at the end of Monday's lecture, but we should finish with both of these by the end of lecture today. And so to get the full story of how it is that genes direct the assembly of proteins, you have to look at the second type of nucleic acid called ribonucleic acid, or RNA. They're both polymers of nucleotides. The differences are, first of all, typically RNA is single-stranded. You can make double helix-shaped RNA, and sometimes you'll get strands of RNA that bond to themselves. So you'll have a kind of hairpin loop structure that may have some double helix parts. But by and large, DNA doesn't form long-term stable double helices. The nucleotides contain the sugar ribose, not the sugar deoxyribose. RNA uses one of the four bases is a little bit different. Instead of thymine, it uses uracil. The adenine, guanine, and cytosine are exactly the same as DNA. And then RNA is found outside the nucleus. If a eukaryotic cell starts leaking DNA out of its nucleus, that eukaryotic cell is dying, if not dead. In a healthy eukaryotic cell, DNA stays right where it is in the nucleus. It's RNA that actually goes out to the places where the proteins get made. So I think we had this last time. On the right is an RNA nucleotide, and you'll notice that right there it's got that extra that says OH, that's an oxygen and a hydrogen stuck to that ring of carbons right there. That's present in ribose. Over here, it's not there. And that's what makes this deoxyribose. And then the other thing is that in RNA, adenine, guanine, and cytosine are the same as they are in DNA. But here in this RNA, instead of thymine, they use a slightly different base that's missing that little methyl group that's missing this. And that makes the base uracil, not thymine. So what happens when a cell needs to make a protein? A length of DNA unzips, it untwists, and those hydrogen bonds that lock the two strands together, those break. Remember that hydrogen bonds are kind of the cell's Velcro. They're easy to break and easy to reform. Other types of chemical bonds are much stronger and take more energy to break. But hydrogen bonds are easy to break, so DNA can easily unzip. And there's a set of enzymes that do this. There's one in particular called RNA polymerase that assembles RNA nucleotides, which are floating around in the nucleus, and assembles them to make a complementary strand to one of the DNA strands. So this follows the old familiar pairing rules. If you have an A on the DNA strand, it will match with a U on the RNA strand. If you have a T on the DNA strand, it will match with a U on, or with an A, sorry, on the RNA strand. And then G on the DNA will pair with a C on the RNA, and C will pair with G. So when the RNA strand is completed, it peels off, and the two DNA strands can come back together and form that double helix again. And that RNA strand is called messenger RNA, which we usually abbreviate as mRNA. And that's a diagram of what it looks like. You can see that DNA is forming our old friend, the double helical structure, And then right here is a zone where that DNA has unzipped and come apart. And what's making DNA do this is an enzyme called RNA polymerase. And the RNA polymerase is doing the work of picking up RNA nucleotides and welding them into a strand of RNA that's forming right here, that's complementary to the DNA, and then peeling off the DNA. So we make a strand of messenger RNA that carries what amounts to the same information as the information on one of the strands of DNA. We're copying information here, and this process is called transcription. So the messenger RNA leaves the nucleus, assuming it's a eukaryotic cell. If it's a prokaryote, a bacterium, or an archaean, all of this is kind of happening in the same big room. If it's a eukaryote, the messenger RNA can pass out of a little tiny holes in the nuclear membrane called the nuclear pores. And they go out to a tiny structure called a ribosome. There are at least millions of ribosomes in a typical eukaryotic cell. I'm not really sure how many they are, but it's a whopping big number. Each one is roughly 20 billionths of a meter long. That's way too small to see with any light microscope. You'll never see ribosomes with light. And each ribosome is made of roughly 50 odd proteins and some RNA molecules that are called ribosomal RNA. We don't really need to worry about what that does specifically. So a ribosome is a protein, a set of proteins that forms a complex with 4 different types of RNA molecules that are specific for that purpose. And we call those ribosomal RNAs. And the messenger RNA makes like Madonna and gets into the groove. That's a computer model of what a ribosome would look like. And it's made of 2 subunits. And this is one. You can see it's kind of colored cyan. So that's one of your ribosomal subunits. And this is the second, the one that tends to be colored more purple. And you will notice that there is a very fine groove into which the messenger RNA can fit. You young post-millennials with your avocado toast may not remember this, but I remember cassette tapes. And to play a cassette, you put the cassette in a cassette player, and the cassette and the player were arranged in such a way that the tape would fit into a groove between the rollers and the magnetic playback heads. That's my personal visual for seeing what RNA does. It threads into the groove in between these 2 subunits. So the messenger RNA makes like Ace Frehley's solo album and gets back in the New York groove. Okay, that's kind of an obscure musical reference for you there. Now what? Well, the ribosome's where protein assembly actually happens. And it happens because of a second type of RNA that you need to know about. I've mentioned messenger RNA. There's RNAs in the ribosomes, and we don't really have to worry about how those work. But the second type that's important for my story is called transfer RNA, or tRNA for short. And a tRNA molecule is a single strand, but it forms several loops, kind of like a hairpin does. The single strand will actually pair bond with itself. And so it forms a set of very narrow loops. One of those loops is called the anticodon loop. And the anticodon loop happens to have 3 bases. sticking out in a prominent position, kind of like buck teeth. It's just the way that the transfer RNA twists is that three bases, a group of three, are sticking out in a prominent position. And then one end of the transfer RNA is bound to a particular amino acid. And here's the thing, which amino acid it is depends on what bases are present in the anticodon loop. So there's a simplified molecule of a transfer RNA molecule, model of a transfer RNA molecule. An actual one, if you could see it, would look a little more twisty than this, but this will give you the general idea. It's a single strand, but there's several places where that strand forms hydrogen bonds with itself, kind of like a piece of tape that you were using to wrap a Christmas present before it started sticking to itself. And the shape is often kind of described as a clover leaf. What's important for our purposes is at this end, we have this anticodon loop. And right here, we've got three bases that stick out in a prominent position. There's other names for the different loops. And then up here at this end, we have an amino acid. To assemble proteins, we have to read nucleotides three at a time. It was first thought that maybe each nucleotide would stand for an amino acid, but in fact, we only have 4 nucleotides, but we have 20 amino acids. It was hypothesized once that maybe every combination of 2 nucleotides stood for a different amino acid, but even that won't work because if we have 4 nucleotides, there are 16 possible combinations of 2. What happens is that you read nucleotides 3 at a time, and a group of 3 nucleotides is called a codon. The analogy I can think of is maybe not one that you're that familiar with, but I'm assuming you've heard of Morse code. Back in the days before, you could send text over, over communications lines with fax machines, and then of course with email and the web and things like that, telegraphers got messages across by using Morse code. There's only 2 symbols in Morse code, a short pulse and a long pulse, usually represented as a dot and a dash. But you can group together groups of dots and dashes to spell out letters and messages of any length that you want to. So if you send out 3 short pulses, dot, 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 that stands for the letter S. And if you send out 3 long pulses, dash, 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 that stands for the letter O. So if your ship is sinking, you send out SOS, dot, 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 and so on. So you can group these 2 basic symbols, dots and dash, but if you read them in groups, you can make different groups of dots and dashes stand for letters, numbers, and symbols. If you've ever seen old movies, there's some war movies where you'll see somebody in a room tapping out on a telegraph key, sending, something like that. That's the best metaphor I can come up with for how this works. Just as you can group dots and dashes together and make different groups stand for letters and numbers, you can group nucleotides in groups of 3, and each group of 3 is a codon that stands for 1 amino acid. Now, of course, there are 64 possible codons, but 20 amino acids, which means that some amino acids, in fact, many of them, probably most, are represented by more than 1 codon. And then of those 64, there are 3 codons that are called stop codons because they don't code for any amino acid. What they do is make the ribosome fall apart and stop assembling the protein. That's how the ribosome, quote, knows when to stop, is when it hits one of these stop codons. So here's a ribosome on the left, and there's a piece of messenger RNA that makes like old flat top and comes grooving up slowly and slips into the groove on the ribosome. Now, this part up here, there's a kind of leader sequence that we don't need to worry about. But right here are the first 3 nucleotides that are actually coding for the protein. As I've said, there's a sequence ahead of that we don't really need to worry about what that does. That's a leader. But right here is the first codon, and it just so happens to be AUG. Now what? There's lots of transfer RNAs that are floating around, and various ones will try to fit into a little slot on the ribosome, kind of like you might have many different keys, and you try every one of them to see if it fits a lock. So this one will go in and see if it fits, and then it falls out, and this one will go in. If it doesn't fit, it'll fall out. And the only one that can fit in the slot and stay in position is the transfer RNA, whose anticodon loop is UAC, because UAC is complementary to AUG. Why does that matter? Thanks to some enzymes called aminoacyltransferases that take care of this, the only transfer RNAs that have UAC on the anticodon loop are the ones that carry an amino acid called methionine. So if it has UAC right here, it has methionine right there. So the transfer RNA in the ribosome essentially read AUG right there. That's the codon. And they make AUG stand for methionine. So the transfer RNAs come in, and the only one that fits is the one that's carrying the amino acid methionine. That's our first amino acid. Now what? Then the messenger RNA moves 3 nucleotides down. It goes ka-chunk, ka-chunk, ka-chunk. And now the next 3 nucleotides are right here, and they happen to be UUU. Me, no, UUU. That's our next codon. Well, all of these different transfer RNAs will try to fit in. They'll pop in the slot and then fall out and pop in and fall out. And the only one that can stay, the only one that can form hydrogen bonds right there, is the one whose anticodon loop is AAA. Must be a Canadian molecule if it's saying AAA all the time. A. Okay, sorry. Canadians say a lot, A. All right. And the only transfer RNAs that have AAA on the anticodon loop happen to be the ones that are carrying the amino acid phenylalanine, abbreviated PAG. So there. If the next 3 nucleotides here, that's the codon, are UUU, the only transfer RNA that can bond with that has AAA on the anticodon loop. And thanks to these enzymes that do the work of putting them together, the only transfer RNA that's got AAA is the transfer RNA that's carrying the amino acid phenylalanine. So that's the second amino acid in the chain. And now a peptide bond forms between the methionine and the phenylalanine, and that first transfer RNA falls off. And eventually it'll get recycled. It will get another methionine stuck onto it. and eventually be reused in making another protein somewhere down the line. Now the ribosome goes ka-chunk, 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 and the messenger RNA moves down another 3 nucleotides, and here's the next codon. It's actually UCA. So yes, little known fact, UCA is one of the few American universities whose name is also a messenger RNA codon. There's a few more. University of Georgia is UGA, and Catholic University of America is CUA. And we're not going to do this anymore because we're changing athletic conferences. But we used to play Abilene Christian University in all of our sports, and they are ACU, and so on. So yeah, there's a few universities whose abbreviations are also codons, and UCA tends to be one. And so we have the codon now is in the ribosome, UCA. Different transfer RNAs will fit into the slot and then maybe fall out if they don't fit. And the only one that fits, that can form hydrogen bonds, is the one whose anticodon loop has AGU, which stands for the American Geophysical Union. It's a society of geologists. I don't know what that means. This UCA is the opposite of the AGU or something. Anyway, the only transfer RNAs that have AGU at the anticodon loop are the ones that have an amino acid called serine. So if anybody ever asks you, what does UCA truly stand for? The answer is it stands for the amino acid serine, which is probably not the answer that they're looking for, but it is absolutely true. So serine comes in, and serine now forms a peptide bond with the phenylalanine, which you might remember has already formed a peptide bond with the methionine. And now this transfer RNA will fall off, and we have methionine, phenylalanine, serine. And now this will fall off, and the ribosome will go ka-chunk, ka-chunk, ka-chunk. And now UAU is going to be the next codon that it reads. And then comes the codon GGG, and then the next codon is CUU, and the next one is GAU. Each one of these groups of 3 can only pair with one of the transfer RNAs, and each transfer RNA is bringing only one specific amino acid. And so the chain of amino acids gets longer and longer and longer. This, by the way, is what the code book looks like. So UCA is, here's a U, there's C, and right there is A. So that's serine. If we want to find out what Abilene Christian University stands for, there's A, C, and then in that box where they intersect, there's U. So Abilene Christian University stands for the amino acid threonine. Sorry, just saw something on the chat. Let me, okay, just somebody entering a name. If we want to know what, what would be a good one? I don't know if there's a University of Colorado at Gunnison, but nah, that one's not interesting. That's also serine. Catholic University of America would be C, U, and then A right there. That's the amino acid leucine. And you'll notice that many of these squares have the same abbreviation in them. That's just because most amino acids have more than one codon that codes for them. So CGU, CGC, CGA, and CGG all code for the amino acid arginine. Or GGU, GGC, GGA, and GGG all code for the amino acid glycine, and so on. But if you look up here, UAA, UAG, and UGA are stop codons. They don't code for an amino acid. They essentially tell the ribosome, we're done. So when the ribosome hits a stop codon, the process comes to an end. The completed protein falls off and goes floating off to wherever it's needed. The ribosome will actually separate into subunits. So the ribosome falls apart. The pieces might get put back together. The messenger RNA might stick around. In bacteria, you'll often have several ribosomes working on one piece of messenger RNA at the same time. In eukaryotes, not so much. But the messenger RNA might be broken down or it might get reused. It all depends. And the protein is now finished. And the protein sometimes has to get chemically modified before you can use it. When that happens, we call it post-translational modification. Ugh. So there. That's how you take the, let me see if I can break this all down. You got one of each chromosome from your mom and one of each chromosome from your dad. Each chromosome is a really, really long length of DNA. Each chromosome bears thousands of sections on it, and those are called genes. Each gene is just a length of DNA, and each chromosome has thousands of genes along its length. Each gene is a length of DNA that has nucleotides in a particular order. The nucleotides in a gene get transcribed onto a piece of messenger RNA, and then the messenger RNA goes out to the ribosomes and is used to direct the assembly of a protein. So what each gene is the instructions for putting amino acids together in one particular order. The order you put amino acids in determines the shape of the protein. If you happen to have inherited instructions for making the protein hemoglobin that replace amino acid number 6, I think what's supposed to be there is glutamic acid, but if you inherit instructions that say put valine as amino acid number 6, what you end up with is a molecule of hemoglobin that doesn't have the right shape and can't carry oxygen properly, and actually causes your blood cells to deform, and you have a condition called sickle cell anemia, for example. That's the ultimate link. That's the, well, it's not the end of the road because there's no such thing, but that's ultimately what Mendel, dithering around with growing pea plants in his monastery in Brno, has actually led us to. That's sometimes called, by the way, the central dogma of molecular biology, in that information in a cell goes in one direction. It's stored in the DNA. The process of transcription makes a piece of RNA using that DNA as a pattern, and that RNA carries the same information as the DNA. And then the messenger RNA is translated, meaning that the order of nucleotides on the messenger RNA is read and used to assemble the amino acids of a particular protein. So again, there's actually exceptions to this, and we'll talk about one in a little bit, probably on Friday. but that's the central dogma of molecular biology. That's the direction that information flows in a cell. Oofta. Okay, I'm actually going to end that early just because that's a good place to end. It's a good place for you to stand on top of this mountain and look around and be amazed at how far you've climbed. And on Friday, I'll talk a bit about viruses and vaccines because, for example, the Pfizer shot that I was able to get back in February is actually an injection of messenger RNA. One of the second one that was introduced, the Moderna vaccine, you're actually getting messenger RNA injected into you. And now hopefully you've got some idea, if you didn't already, as to what that messenger RNA does. It actually tells your cells to start producing a type of protein. But we'll talk more about that on Friday. I'm going to go ahead and stop the recording.